Hi everyone, my name is Paul Boynton. I'm the Assistant Hatchery Manager at the White River National Fish Hatchery in Bethel, Vermont. I'm going to talk today a little bit about some of the research that we're doing there, but before I get there, I'm going to just talk a little bit more generally about the hatchery and what we do briefly, and then we'll focus in a little bit more. Oh, yep, sorry about that. I'll give it a shot. So this is just a map of where the hatchery is located. So right in the middle of the state there is where White River is located, and then we have this other hatchery, Dwight D. Eisenhower, which is our sister hatchery. So we work pretty closely together on a variety of different programs. This is an aerial view of the hatchery. So it's one of the bigger hatcheries in the Northeast region, infrastructure-wise. So we have a lot of capabilities here. These silo-looking things on this picture are outside circular pools, so that's where we keep most of our fish. This is mostly a broodstock facility, which means that we have the big breeding fish. So we produce eggs for other hatcheries, essentially. This hatchery was closed down back in 2011 with Hurricane Irene. We got flooded, and it was closed down for a number of years, and then we just reopened in 2017 with 3 new missions. So those 3 missions, we aid in the restoration of lake trout in the lower Great Lakes here in Ontario. We also help out with conservation of landlocked Atlantic salmon in Lake Champlain and Lake Ontario, and we also provide a space for fisheries-related research at the hatchery. So we've had folks from Concordia, Purdue, Boston University, Dartmouth come and do projects at the hatchery. Actually, Dave, at one point in time, did a research project way back in the day at the hatchery, so that was pretty cool. So those are some of the things that we do. We produce, typically, we'll do about a million green lake trout eggs a year, and then sometimes we do up to 2 million salmon eggs a year. And we distribute those salmon eggs to the Eisenhower Hatchery and the Adirondack State Hatchery, and then we save some at our hatchery to raise up for Lake Ontario as well. I'm going to skip this slide, actually. So getting into the actual research that were conducted. So Lori kind of touched on this earlier, this thiamine deficiency complex issue that we're focused on. And she talked a little bit about, this is not a problem that's just endemic to Lake Champlain. Thiamine deficiency complex has been seen in other parts of the world for quite a while now, actually. So it was first identified in these 3 areas, so the Baltic Sea, Fingers Lake of New York, specifically Cayuga Lake. I don't know if I'm pronouncing that correctly. Hopefully it's spelled correctly. And then also the Great Lakes. And so in the Baltic, they call it M74, or at least they originally did because they identified it back in 1974. They call it near the Fingers Lake Cayuga syndrome because they first identified it in that lake. And then the Great Lakes, they called it early mortality syndrome, which is a really good, I think, name for this issue because that's when these thiamine deficiency complex is impacting the fish. So when the fish first hatch, there's not enough thiamine in their eggs, so they don't have enough thiamine. And as Lori pointed out, thiamine is a really important vitamin B1. It's really important for neurological development, cardiovascular health, metabolic processes, among other things. And everyone needs vitamin B1. We need it, animals need it, fish need it. So it's really important. And so these fish hatch, they don't have enough thiamine, and then you see huge mortality events. So even in a hatchery setting, you typically would like to have a survival of like 80 to 90% survival in a hatchery setting, and they were seeing mortality of 90 to 100%, which is mind-boggling. So they finally identified the culprit as forage fish. So in Lake Champlain, the main culprit is alewife, a non-native species in the lake that were introduced in the early 2000s. And these alewife now make up a pretty good portion of the landlocked Atlantic salmon forage base. And these alewives contain thyminase, which is a thymine-depleting enzyme. So the salmon eat the alewives, take in that thyminase, and it depletes their thymine. And then, again, they can't put enough into their eggs, and so their offspring die. So back when we were first reopening, we were partnering with a PhD student from Purdue, Avril Harder, who a brilliant scientist, who worked with us on this research project. And so for 3 years, we took about 60 families worth of eggs from Lake Champlain, salmon from Lake Champlain. And then each family's worth of eggs we split into 2 groups, as this diagram is showing here. So all these salmon eggs on the left, the first one on the top is from family 1, the one in the middle is from family 2, and the one at the bottom is family 3. And those eggs on the left were put into a bath treatment, a thymine solution, and so they were treated with thymine. And then the other group from each family was not treated as a control, and then we tracked survival. So the families that were treated had really high survival, which we expected because they had enough thymine in them, and then the families that were untreated had poor survival, which we expected, with some exceptions. Some families, for whatever reason, even though they were not treated, had really good survival. And so Avril looked into those fish, their genetics, and she was able to identify just over 1,400 genes that played a role in thymine interactions. And within those 1,400 genes, there was just over 800 that seemingly allowed them to survive with lower levels of thymine. So we kind of ran with that, and I'm actually going to go back. We created, within each year class of fish that we have at the hatchery, we have 2 different groups. We have our max diversity group, which is made up of fish that come from these groups that were treated. So all 60 families of fish were represented, so all those genetics are represented. And then we have a low thymine tolerant group, which come from the fish that were not treated but still survive, so that have these adaptive genes. So Lori talked a little bit about different ways that we're kind of testing this and looking more into this genetics, and this is one of the ways that we're doing it. So we've taken a subset of these different broodstock groups. So we have our max diversity and our low thymine tolerant group, and we pit tag them all so that we can identify who is who, and then we put them together in 2 different pools outside, and then we fed them a low thymine diet that has something called improlium on it, which is just an organic compound that helps reduce thymine in the body. And we fed them that diet for about a year, and then tested thymine levels. And the fish that had been on this experimental feed for about a year had a range of about 5.7 to 3.5 nanomoles per gram. In the lake, we've seen as low as 2.5 nanomoles per gram, and then a fish at the hatchery that's getting the normal diet is around 30 to 48 nanomoles per gram, so much higher levels of thymine. So we are continuing to feed this experimental feed to our fish for another whole year. So by this upcoming November, when we spawn them again, they'll have been eating this food for about 2 years. So we're trying to get that level a little bit closer to what we see in the lake. So once we go to spawn these fish, and the whole idea here is, we're feeding them this low thymine diet. We want to see what their offspring survival is going to look like, right? And what we expect to see is that the eggs that come from that low thymine tolerant group will have good survival, and the eggs that come from the max diversity group will have worse survival. So that's kind of what we're hoping to see. So this is just a diagram of how we will go through the spawning process for this study. So we'll take a female and we'll spawn the eggs, and then we'll take a subsample, about 50 eggs out, just to do some thymine testing on it before we do anything else. And then the rest of those eggs are split up into 4 groups. The first 2 groups, group 1 and group 2, will be fertilized by 1 male, and then the other 2 groups, 3 and 4, will be fertilized by a second male. And that's just to make sure that if we do see mortality, it's not because we had some bad fertilization going on. So we're just trying to control that variable. And then within group 1 and 2, one of those groups of eggs will be treated with thymine. The other one will not be, just as we did before. Same thing with groups 3 and 4. So we're just trying to make sure, if we do treat them, they should survive well, whether they're max diversity or low thymine tolerant. If they're not treated, we hope that the low thymine tolerant group does well and the max diversity group does poorly. These are just some pictures of the actual process this past November. Shout out to Vince, Frankie there, who took these pictures and let us borrow them. But you can see that we have someone with that yellow thing in their hands, which is a pit tag reader. So we're taking a reading of that fish so we can identify what group they come from. And then we're writing that down. We're taking our bowl of eggs, and Kurt here is separating those eggs into these 4 buckets here. And before he does that, in this black tub, we have a scale that's just measuring how many eggs we have, and that's just how we can compare between the different females. And then Lori over here is getting ready to add a solution of thymine into 2 of those buckets. So again, out of those 4 buckets, 2 of them get treated. The other ones do not as a control. And so she's going to add a solution of thymine after the eggs have been fertilized, and we'll wait 2 minutes. And then after that, we'll be bringing them inside. And this is where we house our eggs. So the picture on the left here is just a couple of egg stacks, and we use 2 different egg stacks for this research project. So the egg stack on the left is going to be treated, and the one on the right is going to be untreated. And so all of the eggs, obviously, that are treated are going to go on one side and vice versa on the other. And then this picture on the right is what a tray looks like when we pull it out, and we have these dividers in here. And that's where we're going to actually keep the eggs separate so that we can keep track of them. So we got this little diagram on the left, and that matches up with these cells in the tray on the right. And so in that back of the tray on the left, we're going to have the eggs that come from female 1 that are fertilized by male 1. And then the cell to the right of that is going to be eggs from female 1 that's fertilized by male 2. And again, all of these trays in this one stack will be treated, and then in the next tray, we'll put all the untreated ones. And so from there, the fish will actually hatch in these cells as well. So that red row will actually eventually be empty, and we do that just to make sure the fish aren't swimming between cells, because that would screw up our results, right? And we want to make sure that's not happening. Initially, we do put a small amount of eggs in that row for about 48 hours, and then we'll sample them and take some analysis of those eggs post-treatment to make sure that we did boost them, their thymine levels. But in the literature, we found that you really need to rinse them for 48 hours to get any thymine off the outside of the shell, because we don't really care what's on the outside of the shell. We want to know how much thymine is inside the shell. So eventually, that row will be empty, just as a control. And then we'll track survival. So we'll go in about at least once a week, and we'll pick out any dead ones and keep track of that. So this past year was, like I showed you, our levels, our thymine levels weren't as low as we see in the lake, so we didn't see a whole big die-off that we expected to. And the threshold that's required to induce TDC it kind of fluctuates. So there isn't a number that if you hit it, you're definitely going to see thymine deficiency complex happen in fish. Some fish do better with higher levels, and some fish do better with lower levels. It kind of changes a little bit. So we're going to try this again this upcoming year, have them on the feed for a second year, which in other scientific papers have said that that's a better way to do it anyway. So hopefully we'll be seeing some good results, and maybe we'll be back to talk about them. Now, I don't have a really cool question slide like Lori and David did, but this is the end of the presentation, so if you do have questions, I'll give it my best shot.
So other areas have had this experience with alewives and the effect it's had on the fisheries. whatnot. Is there a period of time when it really affects them? Develops that is resistant, and in the Great Lakes that's been experiencing this longer than here in Lake Champlain?
Yeah, that's a good question. I'm not an expert on the Great Lakes, so I wouldn't want to hazard too much of a guess on that. But I imagine that there have been forage fish that have had this thymidase for just longer than Lake Champlain has, and the whole idea of there might be strains that get it, adaptive to this issue. I mean, that's essentially what we're trying to do, right? We want to breed fish that can reproduce successfully on their own with lower levels of thymine. So rather than trying to remove the non-native species like alewives from the lake, which is incredibly difficult to do, right, and expensive, the management strategy is, can we just adapt the fish to the new reality, right, using their own genetics. And part of that too, like Lori touched on, is to, we want those 800 genes that allow them to do that, but we want to make sure that we're not missing other genes that may be important for their life success, right, or their reproductive success. So there's a lot more checking that we have to do. So this is just kind of the beginning. Thank you.
What the forage fish are actually at. And so that could be part of the cycle, right, is that fish are able to have a very diverse diet, not necessarily just eating alewives. But right now, alewives had taken such a peak that they overcome, like there was more alewives than like smelt. And rainbow smelt was the primary forage base before their introduction. And so there could be cyclical events based on the actual composition of their diet. And so the forage fish study that UVM should be taking on here soon will help to inform like what's going on out here in Lake Champlain. I'm not an expert in the other lake areas, but I do know that so alewife is more of a natural fish, and so there are die-offs of alewife. Temperatures are too high or there's a big temperature swing. And so there is definitely maybe years where there's less alewives abundant, and so it's really dependent on like the age of the fish.
And to add to that too, in the Baltic Sea, landlocked Atlantic salmon and anadromous Atlantic salmon have coexisted alongside alewives for a long time. And so when they started seeing this issue, they were like, well, what the heck? Like, what changed, And the best hypothesis is that the forage base changed. And so alewives became a much bigger part of the salmon's diet. And smelt also do have thymidase in them, but it's much lower levels than alewives. And so they thought that, in the past, they just had enough other fish to be able to supply enough thymine, and now they don't because that forage base has changed. Yeah.
They have noticed, like you said, the alewife population, it really peaked, it dropped, and now the rainbow smelt, they had dropped when alewife came in, and now they're coming back. So the salmon over there are starting to, they're not having quite as many issues with reproduction with the thymidase because of those trends, like you said. So there's probably a paper you can find on the internet somewhere, but just from talking with people and biologists over there, that's, I think, like you said, that's a trend. I don't know if that's going to happen for Lake Champlain. I wouldn't count on it. We've had alewife for a good while now, and the smelt don't seem to be bouncing back. So.
Excuse me. Are salmon more susceptible to the thiamine deficiency problem than lake trout, which seem to be recovering more successfully on the lake?
Yeah, that's a really good question, and I'm not entirely sure. I know that there has been there has been talks about lake trout in the Great Lakes having maybe some issues with TDC, but they're not as concerned as, and they haven't been impacted as much as, for example, salmon in Champlain. So they are seeing species out west too that are having issues with TDC, Pacific salmon, I believe, and I think the actual forage base there has thymidase is anchovies, right? Yep. So it really depends on what region you are, what forage fish you're talking about, and which forage fish have thymidase or not. So, but it is part of the conversation, in my understanding, in the Great Lakes. Cool. Well, thank you so much.